Hello everyone. Hello. How are you all? Yes. So here I am. My name is Bassi and I am your biology educator and this is your second class of photosynthesis, right? So before starting today's class, what I will do is that I'll make you revise some of the things which we have done in the previous class. Okay, will that be fine to everyone? Yes or no? Great, very great. I hope that I am clearly audible and I am clearly visible to each and every one of you. So let's start this session by revising the previous one. Whenever I will be starting this session, I'll revise the previous things very quickly so that you won't have that thing ki you are just learning something from very scratch, right? I understand you are a school student or you might be a dropper. There might be some time in which you haven't got the time for the revision, so let me take care of that thing as well. So in the last class, whatever we have discussed, we discussed about the photosynthesis. What was photosynthesis, right? We discussed the chemical formula, which was C6H12O6 and all those things. We also discussed that photosynthesis was a physiochemical process, right? And if you are the one who just has came to watch this particular lecture, if you are a person who is watching this lecture in recording, after the class, I will add these timestamps so that you can directly jump to the topic. Great. Okay. Now, we discussed about the experiment. The experiment was all about the black paper experiment which we did, right? And this experiment showed that the sunlight is very much necessary for the photosynthesis. Yes or no? Yes, sir. Next, what, sir? We also discussed about the half leaf experiment, which was also known as Mole's leaf experiment, in which we use the KOH. And what was the purpose of KOH? Can anyone tell me? Can anyone tell me in the comment section that what was the purpose of KOH? So, sir, the purpose of KOH was what? The purpose of KOH was to absorb the carbon dioxide gas. Clear? Okay, my voice is pretty much clear and I am clearly audible and visible to you also. Now, we have discussed the important scientists. We discussed about the Stephen Hales, who was known as the father of plant physiology. We also discussed about the father of Indian plant physiology, which was J.C. Bose, and we discussed that Stephen gave about some relationship about the carbon dioxide and the light thing. Great. Now, let's move on and let's talk about the Joseph Priestley. So Joseph Priestley was that person who discovered that bell jar experiment he did with mice and all those things, right? Then it came Jan Ingenhouse. So Jan Ingenhouse used the hydrilla plant to do the experiment. Yes or no? Do you remember this? Then he found a gas bubble, right? Then all of them, that this Ingenhouse, he concluded that the sunlight was necessary for the photosynthesis. Then here it came the Julian von Schack. So Julian von Schack was what? He said that glucose is synthesized and it was stored in the form of starch. Yes, great. Now it came T.W. Engelmann, prism thief, right? He stole the prism from his school and did this experiment. He observed that in blue light and the red light, maximum photosynthesis were taken. And to have a proof, he also used an aerobic bacteria, which was Azotobacter. So Azotobacter bacteria was found on those area where the oxygen were releasing. So oxygen were releasing more. Oxygen were releasing in blue area and the red area, right? Why? Because in blue and red the maximum photosynthesis was taking place. Great. Now let's move on and let us talk about the Neil. So this was the first person who discovered what? Who discovered that the oxygen which is evolved, which is coming out during the photosynthesis, is from what? Is from H2O. And while in green and sulfur bacteria, it was coming from H2S, right? So photosynthesis were of two types, oxygenic and non-oxygenic. Oxygenic, all the green plants were oxygenic, and non-oxygenic was what? Non-oxygenic was performed from the green sulfur bacteria and green and red sulfur bacteria. Clear? Okay. Next we have the Rubin and Carmine, the high five persons. So they did what? They did high five things. He took isotope of oxygen, which is O18, and in this way, he proved that the oxygen which is coming out is not from the carbon dioxide, whether it is from the hydrogen. Clear to everyone? Great. Now, we did all these questions, and now today we are going to go back with our lecture, which is location of photosynthesis. So from here, today's lecture is going to begin. Clear to everyone? Great. Now, what is the location of photosynthesis? So whenever this question is asked, you will be like, obviously, sir, the green leaf. Clear? Okay, great. So let's draw a plant. If in a plant, I'll say that it is a trunk. Again, my drawing is what? My drawing is very famous, so you should respect my drawing. So let's suppose that this is a trunk, and in this trunk or stem, whatever you consider it, leaves are there, the whole plant is there, and let us make some leaves, right? So in this portion, the photosynthesis is taking place. But is it true if I say that any other plant part can also do the photosynthesis? Can I say that? Just question yourself, that can it be said that other than leaves, other part plants can also perform photosynthesis? Yes or no? Yes or no? So my answer to this question is yes, provided that it should contain the pigment, or it should be green in color. For as of now, you remember that it should be in green color. Okay, so there are some stem, there are some parts which are new, which is not very old, which is green in color, so they also can perform the photosynthesis. Clear to everyone? Great, sir. This much is clear, right? Now, should we move forward? Should we move forward if this much thing is clear to you all? Yes, great. Now what? Now let us talk about in which cells the photosynthesis takes place. So there are so many cells in the leaf of the plant, right? The epidermal cells and all those things. But in this plant leaf, there is one cell which is known as what? Which is known as mesophyll cell. There is a cell in this leaf which is known as mesophyll cell, and this mesophyll cell contains what? This mesophyll cell contains the chloroplast. This mesophyll cell contains what? This mesophyll cell contains the chloroplast. Yes or no? Clear to everyone? Okay. Now, this chloroplast is what? This chloroplast is a type of plastid, and this chloroplast contains a very important pigment which is known as what? Which is known as chlorophyll. This chloroplast contains a very important pigment which is known as chlorophyll. Clear? This much is clear? Great. Now, in the mesophyll cells, these chloroplasts are what? These chloroplasts are aligned within their walls. So suppose this is a mesophyll cell, and since it's a plant cell, so it will have cell wall. Yes or no? Obviously, plant cell है तो cell wall तो होगा ही होगा. Plant cell है तो cell wall तो होगा ही होगा, right? Okay. So these chloroplasts are basically what? They are aligned here. They are aligned within their walls. Now, why, sir? There might be a reason, right? If they are aligned into their walls only, so there might be a very good reason. So what is the reason here? The reason here is that to absorb the maximum possible light. The reason here is that to absorb the maximum possible light. Clear? Okay. Now, depending on the condition of light, depending on the light condition, this chloroplast, this chloroplast arranges themselves. So let's suppose that if I talk about, if you like think about that there is a big torch in front of you, or there is a mobile phone and you have switched on its flash. Okay, so when you have switched on the flash, what will happen? Your eyes is gonna close. So why your eyes was closed? Just because your eyes was not able to handle that so much amount of light. So in the similar manner, what is there? The chloroplast also align themselves. The chloroplast also aligns themselves according to the light, right? If the intensity of light is high, they are going to align themselves in different manner. If the intensity of light is something different, so they are going to align themselves in a different manner. So let's see. There are three cases. What's up? One is the intensity of light is very high. The first one is the high light intensity. High light intensity. Okay. The second one will be what? The second one is low light intensity. Okay. Low light intensity. Low light intensity. Clear? And let's have the third one. Let's have the third one as moderate light intensity. Let's have the third one as moderate light intensity. I like this green color. It looks very good in black background, right? Moderate light intensity. Although I have so many colors in the option that myself I get confused sometimes that, oh, which color should I use? But this color is also good. See, green, red, orange, these colors are very good. Okay, let's get back to the lecture. So we have three possibilities: high light intensity, moderate light intensity, and low light intensity. Clear? Okay, sir. So for the high light intensity, what do you think? What do you think? How should this chloroplast should arrange themselves so that they are not damaged, they should receive a normal amount of light. Although the light is very high, the intensity of light is very high, but the chloroplast has to align themselves in such a way that they receive the minimum amount of light. Okay. So let us imagine that this is a chloroplast. Okay. This pencil is a chloroplast. Is it visible to everyone? Okay. If not, I'll find something for you. Just a second. I might have something here. Maybe. Let me find something for you all. Okay. I don't have anything right now. So we have to, okay, we have a paper. We have a paper. Let us understand from this. Okay. So let us suppose that this is the chloroplast. Okay. Let's suppose this is the chloroplast. And if I align chloroplast like this, and the light is coming from this side. Okay. And the light is coming from this side. So what is going to happen? The chloroplast is going to absorb the maximum amount of light. Yes or no? Yes or no? This is the way I placed it, and the light is coming from this side. So the whole portion, this portion, the light is going to be absorbed maximally, right? So what, sir? So what? If we arrange the chloroplast parallel, to the light, then what? Only this surface is going to absorb the light, right? Suppose the light is coming from this side. Suppose the light is coming from this side and light came. Okay. Now what? Only this portion is able to absorb the light. But the rest of the light is what? It is just going like this. Okay. So when the intensity is high, when the light intensity is high, so what happens? The chloroplast align themselves in such a position that they are parallel to the incident light. Okay. So they align themselves in such a position, such a way that they become parallel to the incident light. Okay. And this condition is known as what? This condition is known as parastrophe. Parallel, para. Okay. This condition is known as parastrophe. Clear to everyone? I hope that there is no doubt in this thing. Clear? Great, sir. Okay. Now in moderate, what? The chlorophyll is chill pill. Okay, nothing will happen. Whatever the amount of light is, it's moderate. So we can go anywhere, we can roam here and there. So in moderate, what happens? Anywhere, just randomly distributed. Okay. Randomly distributed. Clear? Randomly distributed. Okay. And if it is randomly distributed, it is known as apostrophes. It is known as what? Apostrophe. Okay, apostrophe. Now, next comes the low light intensity. Next comes what? Next comes the low light intensity. So what is gonna happen in low light intensity? What is gonna happen in low light intensity? So in low light intensity, there is one thing which is going to happen here, is that the chloroplast wants maximum light, right? So the intensity of light is already low and chloroplast need the light. So the chloroplast will try to they will try to absorb maximum light from that low intensity source. So now in this case, the chloroplast is going to align perpendicular themselves to the light, okay, so that the light comes like this and it is able to absorb all of them. Okay, if the light is coming from here, so they are going to align themselves. So this condition is known as what? This condition, hi Sid, how are you? This condition is known as what? It is known as epistrophe. Epistrophe. Okay, this condition is known as epistrophe. Clear to everyone? This condition is known as epistrophe. So this is the three conditions. Now let us understand this with the help of a very quick diagram. So let's suppose this is the chloroplast, one chloroplast. Okay. Next we have the second and next we have the third. Okay. This one is the high light intensity. This one is the moderate light intensity. And this one is the low light intensity. This one is what? This one is the low light intensity. Clear? Okay. In high light intensity, what will happen? Parallel, so parastrophe. So if the light is coming from here, the chloroplast is going to arrange themselves like this. Okay. Moderate anywhere. And low, if the low light is coming, then the chloroplast is going to align themselves like this. Clear to everyone? This is parastrophe, this is apostrophe, and this is epistrophe. Clear? Clear to everyone? I hope that this particular thing is clear. Okay. Any doubts anywhere? Let me know. Any doubts anywhere? Let me know. Clear? Great, sir. Great. Okay. Now let's move on. And let us talk about the structure of the chloroplast. Okay. So we all know that photosynthesis is taking place in the green part of the leaves, green part of the plants. Okay. And in that green parts of the plants, we have a cell organelle which is known as what? Which is known as chloroplast. Now inside the chloroplast, there are so many things. There are so many things and we have to understand it. Because if you are not going to understand the structure of the chloroplast, how you are going to learn about the dark cycle, dark reaction, light reaction, Kelvin cycle, this cycle, that cycle. So this structure is very much necessary. I don't know whether you have learned this or read this in the cell chapter or not, but the thing which I know is that I am here to teach you the whole chapter, and this chapter consists of the structure of the chloroplast. So I am going to cover this thing in a very detail, so that in the coming sessions, you don't have to ask me, sir, what is granum, what was thylakoid, what was lamella, and all those things. Clear? So we are going to discuss this in very detail. So first of all, I have made a basic diagram, but before coming to this diagram, I'll make you understand like how we can make this kind of diagram also, and what are the parts of this chloroplast. We will be coming to this diagram, don't worry. First of all, let us draw a very simple diagram. Clear? Okay, great. So now, if I talk about this particular thing, the structure of chloroplast. Okay, structure of chloroplast. Before that, there is one cell organelle which is known as plastids. There is the plant cell organelle which is known as plastid. Now, what is plastid, sir? It was a cell organelle which is present on only plant cell, and it was of three types: chloroplast, chromoplast, and leucoplast. Remember? So chromoplasm was that kind of plastid which has like a red color, green, blue color, yellow color, right? And the chloroplast was that kind of plastid which were having the green color, right? So this green color plastid we are going to discuss here. Clear to everyone? Great. Now, this plastid is what? This plastid, which is the chloroplast, is having double membrane. It is having what? It is having double membrane. So the first one is what? This green one is the outer membrane. This one is what? This one is the outer membrane. And the inner one is what? The inner one is known as the inner membrane. Clear? Great. Okay. I don't think so that you have any doubts in this. The outer membrane and the inner membrane. Yes or no? Yes, sir. Now, can you see that there is some space in between? Yes. Can you see that there is some space in between them? Do you know what is the name of this particular space? It is present between the two membrane. It is present between the two membrane. So what you are gonna name it? You are going to name it as intermembranal space, obviously. Intermembranal space. So I'm just going to write IMS. Acha. And if you need the notes now, so I have after the class I'll share you a link. That link is of a Google sheet. You have to just click on that link, click there, and all the class link will be present there along with the notes and DPPs. So right now, first class there was no DPPs. Today also I am not going to give you a DPP, but from the next time we are going to give the DPPs also, and you have to solve it daily. Clear to everyone? Great. So now we have this intermembranal space. Clear. Now sir, the main thing is that we don't understand the granum, we don't understand the thylakoid, we don't understand these terms, right? Okay. So think about it. Let's have an example. Let's see, this is a paper. Okay. This is a paper, and I have crumbled this paper and I have crumbled this paper. Okay. See, I have crumbled this paper, I have crumbled this paper. This paper is known as what? This paper is known as thylakoid. Okay. This paper is known as what? This paper is known as thylakoid. Clear? Okay. Clear to everyone? This paper, again I am saying, this paper is known as thylakoid. And this paper is also known as thylakoid. This one is also known as thylakoid. But when I make a pile of them, it is known as what? It is known as granum. When I make a pile of them, it is known as granum. Understood everyone? Clear? So the individual thing, this thing is known as what? This thing is known as, oh, see, I erased it. This thing is known as what? This thing is known as thylakoid. This thing, the single thing is known as thylakoid. If they are arranged in stack-like structure, then we call it as granum. Okay. Now, what is grana, sir? The plural form of granum. Okay. We don't say granas, we say grana. Okay. So the plural form of granum is grana. Clear? Okay, sir. Now one more term, which is stroma lamella. Lamella is what, sir? See, lamella is a membrane, leaf lamella, something like that, a plain structure. Okay, we have read it. But this lamella is present where? It is present in the stroma. This particular thing is known as stroma. So we named this structure as stroma lamella, a membrane-like structure which connects to grana. Is the stroma lamella. Why stroma, sir? Because it is present in the stroma. So I hope that this thing is pretty much clear to everyone. Any doubts in this structure? Let me know. Any doubts in this structure? Let me know in the comment section. No? Great. I hope that you won't get confused any time now, which is stroma, which is granum, which is thylakoid, and which is the other things. Okay, there's some more structure which is present here, which are lipid granules and starch granules and all those things, ribosomes and all those things. Okay, one more thing, I am going to ask you a very basic question in this particular thing. What is the special character of chloroplast? What is the special character of chloroplast? It contains its own DNA, yes or no? It is a semi-autonomous cell organelle, yes or no? The chloroplast is what? chloroplast is a semi-autonomous cell organelle. Sir, what is this semi-autonomous? What is this semi-autonomous? So this semi-autonomous means that it contains its own set of DNA, but this DNA is not that much that it can control everything, but it can control some of the protein machineries, it can make some of the mRNA, and it can do small work. So this is what this condition is known as semi-autonomous condition. And one more cell organelle is there, which is a semi-autonomous one. One more cell organelle is there, which is a semi-autonomous structure. Can anyone tell me in the comment section? Can anyone tell me in the comment section? Yes. So the organelle which is again a semi-autonomous, the other organelle which is a semi-autonomous is basically what? It is the mitochondria. It is mitochondria. Clear? It is the mitochondria. Okay. It also contains a DNA. Clear to everyone? That's very good. Now let's move on. Let's move on and let us talk about the structure of chloroplast. We have done chloroplast alignment. We have done. Now the main thing we are going to discuss in this today's session will be what? Will be the pigments in photosynthesis. So what kind of pigments are involved in photosynthesis? We are going to check that. What kind of pigments is involved in photosynthesis? We are going to see that. So very first thing is that we have chlorophyll. Okay, we know that. So we know the chlorophyll. Now chlorophyll is of few types like chlorophyll A, chlorophyll B, chlorophyll C, and all of this. Okay, but before that there is a sun. We all know sun is there, and from the sun the light is coming, and that light the plant is utilizing. Okay, so sir, in the sun we have ultraviolet also, we have some other wavelength light also. So is the plant able to utilize all the things? No, the plant is only utilizing PAR. Now what is this PAR? It is PAR is what? Photochemically act. I have forgotten P. What's the full form of P? Oh my god. Okay, let's move on. So P was what? P was something photosynthesis active radiation. Okay, something was like this. I have forgotten the P. Photosynthesis active radiation. Okay, and this is what? This is only and only from 400 nanometer, from 400 nanometer to 700 nanometer. It was what? It is only from 400 nanometer to 700 nanometer. Clear? Okay, sir. That means the wavelength above 700 or the wavelength below 400 is not useful for the plants. Clear to everyone? Yes or no? Great, sir. Clear? Okay. So 400 to 700 nanometer, this is the wavelength which plant utilize for their photosynthesis. Now, which type of pigments are present in the plant? See, first of all, we have the chlorophylls. First of all, we have the chlorophylls. Now, what are chlorophylls, sir? Chlorophylls are what? They are green colored pigment. Yes or no? We all know that generally chlorophylls are green colored pigment. Green colored pigment. Now, to work, like if suppose you want to synthesize the chlorophyll, if you want to synthesize the chlorophyll, that plant decided, okay, chlorophyll is very low in my leaf, I want to synthesize the chlorophyll, and it was a night time. Like we overthink, and during the night time, okay, we could have done this, we would have taken that piece of last pizza, right? We all think about these things when we go to sleep. So plant was also trying to sleep just like us, and he had a thought, okay, I can see that number of chlorophyll is very low in my leaf. Then he thought, okay, I'm not able to sleep, let's start the synthesis. But no, the plant can't synthesize chlorophyll in night time. Why? Because the synthesis of chlorophyll requires light. Okay, So light is required for photosynthesis of chlorophyll. Light is required for synthesis. Okay, for synthesizing light is required. Clear to everyone? Great job. Now what? This chlorophyll is soluble in organic solvent. This chlorophyll is soluble in organic solvents. Okay, it is soluble in organic solvents. Clear? Great. Now, types of chlorophyll. How many types of chlorophyll is present? So there are chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D, and chlorophyll E. So these are the types of chlorophyll: A, B, C, D, and E. Clear? Now, if I ask you one question, which of the following pigment, which of the following chlorophyll is what is present maximum is or it is present in maximum amount in the whole plant kingdom? In the whole plant kingdom, which chlorophyll is present in maximum amount? So the answer to this question will be what? It will be chlorophyll A. So chlorophyll A is a universal pigment and it is present in maximum amount, or I can say it is found in all the photosynthetic organisms. So this chlorophyll A is what? It is the universal pigment. It is the universal pigment. Clear to everyone? Right? Are you able to understand the things? Okay. Sid is there and some more students are there. Who are they? You can comment. Clear. So universal pigment is what? Universal pigment is chlorophyll A and this is present in the maximum amount. Now let's move on. We will come to this particular thing, but before that, let's talk about some other pigment, which is chlorophyll B. So chlorophyll B is what? Chlorophyll B is an accessory photosynthetic pigment. Okay, like it's like the main work the chlorophyll A is doing, and chlorophyll B is what? Is just supporting chlorophyll A. So chlorophyll B is what? It is accessory pigment, accessory photosynthetic pigment. It is accessory photosynthetic pigment. Clear? Okay. Now here comes the color. Here comes the color. We should know that which color is having, which pigment is having which color. So if I talk about the chlorophyll A, so chlorophyll A is what? Chlorophyll A is blue-green in color. Chlorophyll A is what? It is blue-green in color. Should I write here? Okay, I'll write here. Chlorophyll A is what? It is blue-green in color. But what about these things? This particular chlorophyll B is having what? Chlorophyll B is having a pigment which is kind of yellowish green. They are what? They are yellowish green. What? Yellowish green. Clear? The pigment is yellowish green in color. Okay, everyone? That's great. Now, where do we can find chlorophyll B? So see, chlorophyll B can be found in green alga. You can find it on euglenoids, etc. Okay, so it is present in green alga. It is present in euglenoids, right? And chlorophyll A is present everywhere. Chlorophyll A is present everywhere. Clear to everyone? Great. Now let's move on and let's talk about some of the basic structure of chlorophyll. Okay, so this chlorophyll is made up of what? It is made up of a huge kind of thing. You can see this and this, right? This and this. Clear? So this part, the huge part is what? It is a porphyrin ring. It is what? It is a porphyrin ring, or you can also say it as porphyrin head. And you don't have to learn this structure. The question from this part is really very less. it's not coming from past few years, but I don't know about today, this year. Already the syllabus is reduced. So there are high chance that question may be put up from these part of the chapters as well. Okay. So this is the porphyrin ring, or you can say porphyrin head, and this is known as the phytol tail. Okay. A tail like structure is present. This you can see. Yes or no? This is tail and this is ring. Now this is fine. Okay. In the middle you can see that there is a metal which is magnesium. And what is the oxidation state of magnesium in this case? Oh, you're not able to see. Dekho, see, I have solution for each and everything for you. This is what? This is a metal which is Mg+2. And what is the oxidation state here? It is +2. Clear? And when if you are good in chemistry, you can figure it out that this is kind of something coordination chemistry or something, right? Let's not go into the chemistry. Let's stick to the biology. Clear to everyone? But this structure is what? This structure is of chlorophyll A. Sir, how can you say this? I can say this by seeing this particular thing. Okay, if CH3 is there, then chlorophyll A. In place of this, if you add this, it will be the structure of chlorophyll B. Clear to everyone? Yes or no? Yes? Great. So now let's move on. If this thing is clear, everything is clear. Now let's go and write the formula for this particular thing. So what is the formula for chlorophyll A? Chlorophyll A formula is what do you know? It is C55 H72 O5 N4 Mg. This is chlorophyll A. What is chlorophyll B, sir? C55 H70 O6 N4 Mg. Obviously, na, we have CH3. Okay, in place of CH3, we have added CHO. So what we have done, we have removed two hydrogen removed, and we have added one extra oxygen. And so easy. And you people are like, sir, we don't remember how to learn this formula for chlorophyll A and chlorophyll B. See, you can solve this type of question by using the help of options. You learn anything else like 5, 5, 7, 2, 5, 4. 5, 5, 7, 2, 5, 4. 5, 5, 7, 2, 5, 4. That is chlorophyll A. And when you have to write the chlorophyll B, subtract 2 hydrogen and add 1 oxygen. It's so simple. You only make things complex for yourself. You are the person who is making things hard, right? 5, 5, 7, 2, 5, 4. हो गया याद? क्या चीज था इसमें? 5, 5, 7, 2, 5, 4. Clear? Great. Now let's move on and then start about the another pigment which is known as carotenoids. So we have what? We had two pigments. One was chlorophyll, which we have already seen. Chlorophyll was chlorophyll A, chlorophyll B, C, D, E. We studied about the chlorophyll A and B in detail, and we talked about the structure of the chlorophyll A and B. Now there is one more pigment which is present, and that pigment is known as carotenoids. That pigments are known as what? Carotenoids. Okay. Now carotenoids are what? Where it is present? Where it is present? It is present inside the chromoplast. It is present inside the chloroplast, and it can also present inside the chlorophyll, inside the chloroplast along with the chlorophyll. So it's not hard and fast that okay, carotenoids will only and only be present in the chromoplast. It can be present in the chloroplast also, but the amount there will be little less as compared to the chromoplast. Clear to everyone? Yes or no? I hope that's clear. Okay. So now if I talk about the carotenoids, they are what? They are again of two types. They are carotenes and xanthophylls. They are what? They are carotenes and xanthophylls. Carotenes is something related to carrot, right? Carotenes is something related to carrot. So if it is something related to carrot, can you tell me that what can be the color of this thing? What's the color of carrot? Blue. Who said blue? Huh? Do you eat blue carrot? Are you getting blue carrot in the market? Is that so? Or have you read biotechnology so much that you are able to create the blue carrot? Huh? No. So carrot color is what? Carrot color is somewhat orange. Orange. What's the color of carrot? Orange. It's orange. It is orange. Clear? So the color of carrot is yellow orange. Yellow orange. Okay. So carotenes are what? Carotenes are yellow orange in color. Again they are of three types: alpha, beta and gamma carotene. Alpha, beta and gamma carotene. Okay. What is the function of carotenoids? See, if I ask that I will make some person stand here in front of me, okay, and let's suppose that I am a very big celebrity teacher. Okay, so if I come to public, I need little protection, right? Just like a Bollywood actress. Okay, so these are my carotenoids. These are what? These are my carotenoids. These are my carotenes. What they are going to do? They are going to protect me. They are going to protect me. So what's the function of these carotenes? Function of these carotenes are basically what? To protect the main pigment. To protect the main pigment. How, sir? If suppose the main pigment is standing like this and suddenly a huge intensity or some very huge intensity light came and that main pigment is gone. So what these kind of structure do? They absorb some of the light intensity and slowly they give to the main pigment. Okay, so that the main pigment is still pale and he doesn't get a heart attack by accepting lots and lots of light energy at once. Okay, so the light energy, they help in the energy distribution to the main pigment. Okay, so can I write here? It protects what? It protects the main pigment. Now what is main pigment, sir? Main pigment is basically what? Chlorophyll. Yes or no? Protects the main pigment. The main pigment. With whom? With whom? With highlight energy. And if highlight energy is there, what will happen? Oxidation can take place, right? So we can write it protects the main pigment from oxidation. From oxidation. Clear? Okay. Great. Now next is what? Next we have xanthophylls. Next we have xanthophylls. So what does the xanthophylls is? Xanthophylls is yellow colored pigment. Xanthophylls is yellow colored pigment. Okay, yellow colored pigment. Yellow orange is what? Yellow orange is carotenoid. Yellow orange is what? Yellow orange is carotenoid, and only yellow is xanthophylls. Clear? Great, sir. Now let's move on and let's talk about the next one, which is phycobilins. Okay, there is one more pigment which I am going to discuss here, which is phycobilins. Okay, phycobilins. So this phycobilins are that kind of pigment in which if you pour hot water, they are gonna dissolve. So phycobilins are that kind of pigment. If we pour hot water, they are going to dissolve. So can I write here? They are soluble in hot water. Right? Hot water. Now the time winter is coming and we will be able to know the importance of hot water very soon, right? Okay, now they lack what? They doesn't contain Mg. Mg is not present in them, and they also lack phytol tail. They also lack phytol tail. Clear to everyone? They doesn't contains Mg and they also lack the phytol tail. Yes or no? Great, sir. Now let's move on, and next there are types of phycobilins which can be blue in color, which can be red in color, and here also we have light blue in color. Okay, you don't have to remember the names of this, just Remember, phycobilins are water soluble pigments. What kind of water soluble pigments are? Hot water soluble pigments. Clear? Okay. Now, let's talk about the absorption spectrum, which is again a very important part. So whatever we have discussed in this, the questions can't be conceptual, right? They can be fact based or they can be memory based. If they ask about you that say the formula for the chlorophyll, so you don't have to, apply some concepts. Okay, carbon made a bond, nitrogen, magnesium, no something like that. Either you are able to memorize it, then you are going to answer. If not, then you have to choose A, B, C, D, then you can do akkada something like that. Okay, so this part was very much rote learning, memorizing thing, but now the part is coming where the concepts are required. So if the concepts are required, then we have to study it very well. So what is absorption spectrum, sir? The very first thing, what is absorption spectrum? Suppose this is a chloroplast. Okay, suppose this particular structure is a chloroplast and I am giving light to this particular chloroplast. Now inside this chloroplast we have chlorophyll, and how this chlorophyll is absorbing the light, how the photosynthesis is happening in red light, how the photosynthesis is happening in violet light, how the photosynthesis is happening in yellow light, all these things, along with in which light this chlorophyll is maximum active, in which particular light the chlorophyll is able to absorb maximum light. So all these things is related to this absorption spectrum. Okay everyone, great. So now this is the absorption spectrum, and I hope so that it is clearly visible to everyone. Should I stand here? Okay, I will stand little here, so then it will be clearly visible to everyone. Okay, so this is the absorption spectrum. Here it is what, if I talk about this graph, so this graph is basically what? This graph is absorption of light. This shows absorption of light by chloroplast. Okay, this shows absorption of light by chloroplast. And here we have what? We have the wavelength. Here we have the wavelength, which is lambda, and this is the absorption. I'll write here. I will write here. So X represents what? Absorption of light by chloroplast pigment, okay, absorption of light by chloroplast pigment. Clear to everyone? Great, sir. Absorption of light by chloroplast pigment. Okay, now first of all, how you can learn this graph? Usually we have confusion which one is chlorophyll A, which one is chlorophyll B, and all those things. Okay, so the first one, the first peak is chlorophyll A. The first peak is chlorophyll A. The second peak is chlorophyll B. And the third peak is carotenoids. Simple. Okay, no rocket science, no concept right now. The only thing which you have to remember is that A for apple, B for ball, and C for cat. So A for chlorophyll A, B for chlorophyll B, and C for carotenoids. So which comes first? Chlorophyll A comes first. So here we have chlorophyll A. Chlorophyll B second, and carotenoids, which is third. Clear? So can I say that if this is the 400 and this is the 700, if I am talking about the wavelengths, this is 400 and this is 700. So which means what? This basically represents the red light. This basically represents the red light, and this basically represents what? The violet light. Yes, the violet one. So now where the maximum photosynthesis is happening? Where the maximum photosynthesis is happening? See, maximum photosynthesis is happening in this region and in this region. There are two regions in which maximum photosynthesis is happening, which is this and this. So maximum photosynthesis is happening in red region, and what? And it is happening in where it has gone. Sometimes iPad play games with me, right? Okay, okay, we have this. So now, if I talk about the pigments, photosynthesis. So maximum light is absorbed what? Maximum light of red and red and blue light. Red and blue light is absorbed maximum. Light is absorbed. Yes or no? This chlorophyll A and chlorophyll, you can see the peak, this and this. The maximum peak is in the blue region and in the red region. So I can say that during red and blue light, maximum they are absorbing. Maximum red and blue light is absorbed. Clear to everyone? Okay, sir. Now what? Now if I talk about the rate of photosynthesis. So what is this rate of photosynthesis? See, we saw that the chlorophyll A is active in red region and the blue region. Similarly, the chlorophyll B is somewhat also active in the red region. And now, if I ask you that in which region the maximum photosynthesis will take place, obviously the region in which maximum light is absorbed. So this is what? This is the graph for the rate of photosynthesis. Again, here we have 400 nanometer and here we have 700 nanometer. So I can see two peaks, one here and one here. So these two peaks represents what? This represents the red light and this represents the what? Blue light. Obviously, one represents the red light and the other represents the blue light. Clear to everyone? That's great, sir. Now what? Now, this is the action spectrum. Now let's talk about the action spectrum. How can we talk about the action spectrum? We are going to overlap both the graphs. We are going to overlap both the graphs and we are going to check that what is this graph matching or not. See, I'll show you how. I'll show this very beautifully. Where it is going? Be here. Now let's paste it. So if I overlap this graph somehow, so can you see that we are getting peaks here only, no? Here was one peak, this region, and this region was one peak. So that means the photosynthesis is happening more in that region where the blue and the red light is being absorbed more. So in presence of blue light and red light, maximum photosynthesis takes place. This was all about the graph. Clear to everyone? And you will be like, sir, we are not able to understand the graph properly. We don't know what to do, what not to do. This was it. Just simple. I told you that you are making things complex for yourself. Just take it as it's nothing and you will be able to understand everything very clearly. Okay? That's very good. Now, we have completed this thing and let's go with some of the questions. Okay, let's solve some of the questions for today. So the question says that anthocyanin is present in. Anthocyanin is present in. Is it present in chloroplast? Is it present in chromoplast? Is it present in vacuoplast? Or is it present in vacuole? What could be your answer? What could be your answer? Anyone? What could be the answer? Yes. The answer will be what? The answer to this question will be vacuole. Why? It's not present in chloroplast, neither it is present in chromoplast. Anthocyanin is a pigment which is present in the vacuole. And there is no such thing vacuoplast. And if you have made this answer, na, see, seriously, if you have like used to I thought that okay, vacuoplast can also be something. No, it's nothing. So just ignore the option C. Clear? Now, some of them can also answer, sir, E is the option, right? Production of glucose was discovered by. Production of glucose was discovered by. So in photosynthesis, production of glucose was discovered by Julian Sachs, right? Julian Sachs. Clear? Okay. Now, to discover the site of oxygen release, dash is used. The questions are from the previous class. The questions are from the previous class. To discover the site of oxygen release, which bacteria, which virus or what was used? Anabenna, nostoc, azotobacter, none. Anabenna, nostoc, azotobacter, none. So, sir, Anabenna was not used, neither nostoc, neither none. So, the answer will be azotobacter. So, the bacteria was used was azotobacter. Clear? Okay. Now, did I say something wrong? Azotobacter is the correct answer. Now, which is the most abundant plant pigment? Which is the most abundant plant pigment? A, B, C, D. First, most abundant plant pigment A, B, C, D. Obviously, the most abundant plant pigment is what? It is chlorophyll A. It is chlorophyll A. Next, we have, okay, we don't have anything. We don't have anything next. So, next what? I am going to give you a question. If you don't have anything, you all have me, so I'm going to give you a question. First, let me write the option, then I'll frame the question. So, we have blue light, we have red light, we have green light. Let's make a traffic light. So, let's add the yellow light also. Okay. It should be like red, yellow, green. Okay. Now, among which of this light maximum peak of chlorophyll A is absorbed? Maximum peak of chlorophyll A is observed. Yes. Maximum peak of chlorophyll A is observed in blue light, red light, green light, yellow light. So, it is observed in blue light. Obviously, check the graph. Check the graph, people. See, this was what? This was chlorophyll B. This is chlorophyll A. So, in chlorophyll A, maximum is what? Maximum peak of chlorophyll A is absorbed in the blue region. Clear to everyone? Great. So, I hope that you had a wonderful session, and if you need the notes for this session, check the comment box. The notes will be uploaded there. What you have to do is that you have to click on that Excel sheet, and there you will be able to see the date and the lecture name along with the class link and along with the notes. So till then, bye-bye everyone. See you in the next class tomorrow at 4 p.m. Don't forget to subscribe to this channel and share this among your friends. Thank you everyone.